Hey, good morning. We have a real treat here today. We have Dr. Howard Fields from UCSF who's joining us. I'm going to paraphrase a little bit from his bio and shorten it because it's just it's an incredible career. He is professor and director of the Wheeler Center for Neurobiology, a physician at the University of California, San Francisco. He received his MD and his PhD 52 years ago here at Stanford in 1965. He's absolutely incredible, 52 years ago. He did internal medicine training at Bellevue, a research neurologist at Walter Reed. He then studied neurology at Harvard, and then subsequently joined the faculty at UCSF, where he's been ever since. His major research has been in neurobiology of pain and substance abuse with a focus on how endogenous opioids contribute to reward and pain modulation. He founded the UCSF Pain Clinic. He was just telling me a little bit about how he and Ron Miller put that together. He is the first to demonstrate the clinical effectiveness of opioids for neuropathic pain and topical lidocaine for post-traumatic neuralgia. He worked with Alan Bassbaum, also up at UCSF, discovering a pain modulatory circuit required for opioids that can both produce analgesia and show that descending pain modulation can either inhibit or enhance pain. Working with John Levine, he published his landmark study on placebo analgesia and showed that it's mediated by endogenous opioids. His group recently discovered a novel opioid action related to reward in nerve cells in the striatum and can encode the magnitude of reward. In 1997, he was elected to the National Academy of Medicine. In 2010, the American Academy of Arts and Sciences. That's his bio. That's his brief bio. I will tell you that Howard is a living legend in the field of pain. Truly, I'm just pleased he's left a little bit of work for the rest of us to still figure out. He's made a truly seminal contribution to the field of opioids and reward and addiction and so many other areas that connect. I think at least as importantly, what's not listed here is the number of researchers whose lives he's impacted, young investigators who have grown up under his tutelage and gone on to have successful careers of their own. As you'll find, he's an incredibly warm, engaging, articulate, brilliant man. And he's been gracious to come down and share kind of his overall view in opioids, sharing some of his work and others. So with that, please do give a warm welcome to Dr. Howard Fields.
Yeah, you'll leave that down there. Thanks, Sean. Thank you all for appearing. Can you hear me okay in the back? Thank you all for appearing at this ungodly hour for grand rounds. I'm a neurologist, and we're generally not awake at this time. So if I sound like I'm going a little slowly, you'll forgive me for that. So today, I want to talk a little bit about opioids. Opioids are in the news. Opioids are in the news generally because the media and the public are afraid.
We may need to
move it up.
We haven't put it. Let's get you this guy here.
Is that better? Okay.
You just need to put on the other mic too. That one's fine.
You know what they've got is it's two mics. One they're streaming and recording, and the other one is for us.
Okay.
So I'm the one who didn't follow the rules here.
He told me to get it from you, but he didn't tell me what to do with it. Okay, good. So I want to, what I hope to do in this lecture is clear up some misunderstandings that have been promoted largely by the public media. Can you turn down the lights that are on the screen? It'd be easier to see these. Yeah, a lot easier to see slides. Is that possible?
Yeah.
Okay. So, the story of my career, that's much better. Thank you. The story of my career has been the quest for what I call the holy grail of analgesia, which is a drug that would completely relieve severe pain of all types, that you would have multiple routes of administration, that you could deliver the drug close to where it's needed. Generally, if rapid onset would be great because it gives you complete control, something like remifentanil. and rapid reversibility, which we have with naloxone, and to have no effects that you don't want. Okay, so that's the holy grail. And I can tell you right at the beginning is we are making progress toward having such a drug. And I'm very excited. I think over the next 10 years, opioids are going to become much safer, and they're going to become more useful. Okay, so I always like to do a little audience participation, especially when it's early in the morning. So I want to get some information from you and just ask, how many people in the room have actually ever used opioids clinically? And show of hands? All right. That's reassuring. And how many here have ever used peripherally restricted antagonists for the treatment of those? Okay, I'm glad that my alma mater is keeping up with the times. That's great. How many people have used opioids intrathecally or spinally? Okay, that's great. How many here have used them for the treatment of chronic non-malignant pain? Okay. This is an enlightened group, I have to say. And you'll all be very interested to have hear what I have to say. How many people who use chronic opioids for non-malignant pain worry when they prescribe them? Okay, great. Well, we're on the same page. Okay. So let's go. What I want to do now is tell you why you use opioids, why they're so great. They really are amazing when you think about it. They are effective for severe pain of any type, including neuropathic pain, despite past debates about that. So they're a broad-spectrum analgesia drug. They have a rapid onset depending on how they're delivered and which ones you use. There are multiple routes of administration. People have even used them locally, injected them into joints. Christoph Stein in Germany. Has anybody ever used opiates for local pain? Okay, good. So there's a little skepticism here on that, as there is up in San Francisco, but it's been reported. And I think the key thing about opioids is, compared to general anesthetics, you can get a lot of pain relief while patients are still awake, and that's really the key. So there's something about what these drugs do and how they work that makes them highly specific for analgesia. And most of the time, you can get good analgesia without significant respiratory depression or sedation. Not always, but often. So they're really good drugs. Maybe not ideal, but good. And then, in addition, there's a bonus for many patients. They not only get pain relief, but feel great with the drug. Has anybody here had a patient report that to them? Yeah, okay. And I would say that's a good thing. People like to feel good. All right, so why are they so effective? Well, opiates have multiple targets in the central and peripheral nervous system. And I don't need to list these, but I will show you on these diagrams. It comes from an old review article that I wrote. This isn't projecting too well, so I'm going to walk over here. They work on primary afferents. These primary afferents express mu and delta opioid receptors on their terminals, and the opiates prevent the release of the neurotransmitter glutamate. They also act on second-order cells in the spinal cord to hyperpolarize them and inhibit them. They also work in the thalamus. They also have cortical sites of action, right? So this is the ascending pathway, and opiates work on multiple sites in the ascending pathway. They also, this is the ascending pathway, this is the descending pathway. Opiates work in the amygdala, hypothalamus, midbrain, periaqueduct, brain, and medulla. So they work on the system that is sitting on top of the dorsal horn and controlling the volume of the pain transmission pathway. So you have dorsal horn, you have thalamus, you have cortex, you have hypothalamus, you have midbrain, you have medulla, you have the primary axis. Multiple targets. And that's going to turn out to be very important when it comes to developing new better opioids. These actions at these various sites are not just additive, they can be multiplicative. So you can give a relatively small concentration of an opioid, and because it acts at multiple sites, and because these sites interact, you get a very big analgesic effect, and it's the location of the receptor on specific circuits in the central nervous system that gives it the specificity. Now, unfortunately, there are other places in the central nervous system that aren't so helpful, like the respiratory centers, and that these lead to side effects. So then we get to problems. Sedation. Sometimes sedation's not a bad thing. Did you? Okay. Are we good? Okay. So these are the things you all know about them, so I don't have to belabor them. If we could get rid of those, we'd have better drugs. Okay. So the question before us is, can we increase the analgesic effect and or reduce these unwanted effects? So I want to go a little bit over the history of opioids. The first sort of major step forward after their serendipitous discovery in the juice of the opium poppy was that once pharmaceutical companies started messing with the molecules, so they purified morphine, and then they started putting side chains on the morphine molecule to see if they could get rid of some of these adverse side effects. So they do structure-activity relations, then they put a side chain on and do the same thing with a new drug. What they found was that if you looked at a couple different tests, like here, the potency for relieving pain in people, and here to inhibit gut motility, this is guinea pig ileum, which was a standard model for the mu-opioid receptor. What they found, and this is very important, was that the rank order of potency for a variety of opioids was identical, for analgesia and for inhibiting the gut. The conclusion from that was that there's this one binding site for the mu-opioid receptor, and everything it did depended on its affinity and potency at that binding site. So that was how the mu receptor was defined, and everything was compared to morphine. That was the standard drug. And that's why it's called the mu-opioid receptor. Mu is for morphine. A very cool result, which they got when they stuck on a particular side chain, extended this nitrogen in this ring, and they found that they got a very selective antagonist. So they could take this drug and just add on a couple of carbons with a double bond, and all of a sudden they got a drug that had no analgesic effect. In fact, it would block the receptor at very low concentrations. So this was a huge advance. Now you had reversibility of the opioid, but you also had a way to investigate how opioids work by injecting antagonists into different parts of the brain. So this was a huge event. This was sort of what I would call the defining event, for the mu-opioid receptor, and that's the one we're interested in. That was back in the 70s. Now, since the 70s, made a lot of progress. I would say the first big advance from the 70s is, here is 1992. The first opioid receptor was cloned. So we got the gene. That was the delta opioid receptor. And then very rapidly, within a year, they had 3 other opioid receptors, and the reason that they were able to clone these other receptors, the delta, the kappa, and the orphan receptor, was these little yellow beads. Each represents an amino acid that's identical for all the opioid receptors. They're a very closely related family, and they all have similar cellular effects. The range of cellular effects are different for each of those different receptors. They're most different in their outside chain, which is the nitrogen chain, and the carboxyl chain is inside the cell. These are where the amino acids are most different, so their binding sites and their signaling sites are very different. But their transmembrane sites are very similar. That turns out to be important for something I'm going to talk about later. So, once they had the amino acid sequence, they got the gene. Once they got the gene, they made a mouse. And the most interesting mouse here was reported by Birgit Kiefer about 7 years after they cloned the mu receptor. So she made a mouse that had a non-functional mu receptor, and this mouse, if you gave it morphine, nothing happened. So there was no sedation, there was no analgesia. They didn't like the drug. They wouldn't self-administer the drug. It didn't produce anything rewarding, but there was no constipation, no respiratory depression, right? So this was a pretty discouraging mouse in a way. In one way, okay, it's great. We found the molecular target. We know the gene that underlies everything morphine does. If you don't have that gene, morphine doesn't work. On the other hand, it says, well, if you make a drug that's acting at that receptor, it's going to do everything. So the whole idea that you might, say, have a delta receptor or a kappa receptor or an orphanin receptor, and it would be a better analgesic, or it would be an analgesic without side effects, that kind of went away with this mouse. Okay, so that was in the 90s. Now, not too long ago, and not very far from here, Brian Kabilka's group actually got the first crystal structure of an opioid receptor. And that turns out to be incredibly important in terms of drug design. And I'll talk a little bit about that later in this talk. Okay, so now we're stuck. You take out the mu receptor, you get rid of everything. The whole idea of maybe making a better morphine is called into question. Is there a way to get around that? And it turns out that, in fact, there is a way to get around it, and that's because we now know a lot about the cell biology of opioids. So if you have this is to illustrate the 7 transmembrane domains of a G protein coupled receptor. They're all that way. All the opioid receptors are 7 transmembrane domain receptors. One of the key signaling methods is through G proteins. These G proteins, they can open a potassium channel and inhibit neurons. They can inhibit a voltage-dependent calcium channel and inhibit the release of neurotransmitter. So those were the first things to be discovered. So opioids are largely inhibitory in the central nervous system, but we all know that they inhibit adenylate cyclase, which is an intracellular mechanism. They also activate the MAP kinase cascade, and as you'll see, there's some other signaling, intracellular signaling chains. So now, all of a sudden, if we could selectively activate different downstream signaling systems, it's possible that we could, promote some of the functions of the opioids and inhibit others. And that's been a very active process, and the most exciting recent advances have come from a company called Trevena. I've never consulted for them, so there's no conflict of interest, but they were very interested in this idea that signaling can be either through the G protein or this other protein called beta-arrestin. Beta-arrestin's interesting because when it's activated along with a couple of other proteins, the receptors are internalized and inactivated. But it turns out that beta-arrestin also signals, and as you'll see, because it's signaling differently in different neurons, you can selectively enhance certain biological effects. So what they did was they tried to find ligands which produced a conformational change in the receptor such that it mostly signaled through G proteins and did very little signaling through the beta-arrestin. Right? So they suppressed beta-arrestin signaling in their mu-active compound. And what they found, I think, is pretty amazing. And that's shown in this slide. This is work in human volunteers using an experimental pain model. And what they found, so morphine is orange. That's always the gold standard. And this is the amount of analgesia. The higher up it is, the more analgesic it is. So here's morphine, and this is 10 milligrams. I assume that it's intramuscular. The Trevena compound, middle dose, seems to actually be a better analgesic. But the more interesting thing is over here, this is respiratory depression. So we know that analgesic doses of most opioids will suppress respiration by a certain amount, get an increase in PCO2. Here's the morphine compound, and here's the Trevena compound. So the basic point here is you got more analgesia and you have less respiratory depression, and this is in humans. So this is an incredibly important advance, okay? And it's already, I don't know if it's in clinical trials now, but to be able to have a compound that produces much less respiratory depression, that's got to be a good thing. It also is a proof of principle, right? It says you can manipulate an opioid molecule and improve its sort of profile of benefit in terms of analgesia to cost in terms of respiratory depression. Okay, so any questions about that? Because I think this is really an important point. The other important point that you could draw from this, one conclusion you could draw from this is that we know the circuit in the brainstem that controls respiration in the medulla, it's a different set of neurons than the neurons that are activated to get analgesia or the neurons that are inhibited to get analgesia. So what that says is the same ligand working on two different nerve cells can activate different downstream signaling systems and therefore give you a different ratio of activation, right? So that's the proof of principle. So in theory, if it turns out that the mu receptor on different circuits in the brain, right, has a different ratio of the downstream signaling systems based on, a given ligand, a different ligand will activate different circuits to different degrees, right? So it reopens the question that seemed to be closed with the mu knockout mouse, where all functions were lost. Okay. So the other thing I want to kind of briefly talk about is this protein-protein interaction. This is different. So it turns out that in the plasma membrane, G proteins in particular will often line up together and function as a unit. We call those heterodimers if they're different. We call them homodimers if they're the same. So two mu receptors would come together and form a homodimer. A mu and a delta receptor would be a heterodimer. So the question is there any evidence for heterodimerization? Because if there is heterodimerization, you could give bivalent compounds and get around the issue of a single molecule. So there is some evidence. I'm not going to go into much of it, but there's one paper that I thought was pretty cool, and this is Phil Portuguese's work, University of Minnesota. So he's a pharmaceutical chemist, and there's this big literature that if you give a delta opioid receptor antagonist, it can enhance the analgesic effect of a mu agonist and prevent the development of tolerance and dependence. So that was in the literature using antagonists and delta knockouts, mice. The mu agonists seemed to work better. And so what Phil did was he made these bivalent compounds that had a mu receptor agonist at one end, like a morphine-like drug, and at the other end there was a delta antagonist. So he called these bivalent compounds. But the elegant thing about his work is that he put the mu agonist at one end, he put the delta antagonist based on naltrindol at the other end, and then he separated them by various carbon molecule spacers. And what he found was pretty incredible, that if he got just the right spacing, there's a lot of data on the slide, but the main thing is where the red lines are, that the analgesia was as good as morphine or oxymorphone, which is shown here. This is just the ED50 for analgesia. It's virtually identical. But if you look at a measure of physical dependence, it's like orders of magnitude less, from this bivalent compound, but only when the spacing was just right. So the two molecules have to be reasonably close to each other, within angstroms of each other. So this is another possibility for a molecular modification of an opioid agonist to get around some of the unwanted side effects. All right, where do we go from here? So everybody's probably familiar with many of the compounds shown here. Everybody's used fentanyl. Okay. People have used morphine. So these are different classes of drug. There's the benzomorphins, which include drugs that are used that are peripherally restricted by and large, that are used for the treatment of diarrhea. Imodium, for example, is in this category. So there's a variety of different molecules, and they haven't really been systematically studied for the ratio, say, of respiratory depression, to analgesia. They're all just kind of thrown into a bag of mu agonists. So that's a possibility. Buprenorphine's an interesting drug. How many people here have used it? Okay, so I highly recommend buprenorphine. The reason I recommend it is it's a partial agonist at the mu receptor, so there's reduced respiratory depression just from the fact that it's a partial agonist. Right, but the other thing is it's a kappa antagonist. And as a kappa antagonist, there's growing evidence that can be used to treat depression and anxiety and reduce the intensity of drug withdrawal. And that's what I would call a bivalent single molecule. So it's kind of cool. I think it's underutilized for pain, for example. Okay, so these are some of the approaches we can use. We could just go back and look at all the drugs we have and see how they do on a panel of desirable and undesirable side effects. I don't think anybody's going to do that because it's expensive. You'd have to do it in people, and the drugs are already generic, so there's no money in it. But it could save people a lot of money because the drugs are already out there, and will allow you to know what the optimal drug is to use for whatever purpose you want to use it for. So here's another, really important advance that just really happened, and it happened here at Stanford, and it happened as a result of having the crystal structure of morphine. So one of the things you can do if you have the crystal structure of morphine is you can make a hypothesis of how the molecule actually docks at the binding site in the neurotransmitter, in the receptor Okay, so you got this crystal structure, and you know the side chains for all the amino acids. You can make a hypothesis about what are the lowest free energies, how the opioid sits in the receptor, and then you can say, okay, I've got a series of compounds that have certain properties. I've shown how they dock on the crystal. Now I'm going to do structure-activity relations specifically designed to improve certain areas of binding and to reduce other areas of binding. So now you've got really a strategy going forward for what it is you want to do to a molecule to make it better. This is an incredible advance, and they were able to do this with the mu receptor and come out with a compound that had less respiratory depression, similar to the Trevena compound. So, that's a great step forward. People here who are doing it, if you want to have more details, get them to do grand rounds. Okay. So, things are encouraging, right? We've made progress. We can, deal with constipation because we got a peripherally acting antagonist. That's good. Looks like we've dealt with respiratory depression by directing the signaling away from beta-arrestin. And it looks like the bivalent drugs are effective maybe in reducing dependence and tolerance. Okay. So what about addiction? That seems to be the big problem now. That's what people are worried about. And, first thing I like to talk about is what's the risk of giving somebody an opioid analgesic? How many people here are worried when they use it consistently? Okay, good. I think it's a reasonable worry. However, this is a paper that was recently published by Sean and colleagues in JAMA Internal Medicine. This is a really huge study, an ideal study, I would say. And I'm not just saying that because I'm not numbered here. I think it really is a great study. They looked at, I'm going to say, 18 million sort of people looking at insurance records just to see if, people have gone to the drugstore and filled a prescription for an opioid. And what's cool about the study is they selected people out who hadn't used opioids for a year and then had something happen to them or didn't have something happen to them and then followed them for another year. So they picked it was a 3-year period for every subject in the study, and it was a lot of subjects in the study. And what they found is, yes, after a year, people, if you just follow them, there's a small fraction, around 1% overall, of people who will be on opioids for at least 3 months in a year. And if they've had a surgical procedure, that can increase the probability that they'll be on opioids for that period of time. It depends on the operation. This was a total knee replacement. That seemed to be the one that was the biggest risk, but it's still only 5% of people. And I should say that they're on analgesics, but they're not necessarily addicted to it. They're just getting a prescription refilled. They're still on it. And for most of those people, they're probably on it because they still have pain. The point is, yes, there's a real risk, and the risk is actually pretty small because we don't really even know of the 5% who were still on them, how many of those people were really abusing the drugs. My guess is 10% at most. So we're talking about relatively low levels of risk of having people on opiates, especially if used for postoperative pain. So bottom line, if you want to worry, it's okay, but don't worry too much. All right. So what about addiction? What's going on? And is there something we can really do about it? And this is the kind of work that we're doing now up in my laboratory. So Occasional recreational use, a lot of people use opioids for fun. Then maybe they'll use them regularly, then maybe they'll use them heavily. Sometimes they have a pain complaint. Sometimes the pain complaint is really just a way to get a drug. With regular heavy use, you can become tolerant to the drug, or at least you escalate your dose. That generally means that you've become tolerant, because why would you spend more money if the drug was still working as well as it was when you first tried it? Abuse is partly a social definition. By definition, if you use a drug that's illegal, it's abuse, whether you're using it recreationally or not. With alcohol, you can have 4 or 5 drinks. It's not necessarily abuse, but if you get in a car and you drive and you have a DUI, then by definition it's abuse and you can go to jail for it. So abuse happens. It's way more common than addiction. Addiction means you want to quit and you can't. It basically means you've lost control of your life. Now the drug's controlling your life, and everything you do is in the service of getting the drug, and paying for it. And then a lot of bad things happen, which I don't really need to go into. You know what I'm talking about. So how are we going to go about figuring out, why over time a drug like an opioid can become addicting? Well, the way we've approached it is you decide what the behavior is. It could be self-administration. That's probably a good one because you can train animals to self-administer opioids. They like it. Once you've done that, you find the circuit that's driving the behavior. You find the synapse in that circuit where the drug's working. You find the proteins that are involved, and then the genes. Right? So our focus has been, finding the circuit, defining the circuit, and looking at the synapses. That's what we do. And one of the places that we've been focused on is the ventral tegmental area. Now, I'd like everybody to just say that word, ventral tegmental area. Okay, now you can go and impress your friends. I'm just saying, did you know that opiates produce addiction by acting on the ventral tegmental area? I think, that could be an impressive bit of information. You can check out and see if I'm right. Just walk down the hall and ask 30 people if they know what the ventral tegmental area is. Okay. So the reason that we focused on this is that if you inactivate the mu receptor in the cells in the ventral tegmental area, it's no longer rewarding. So you can pick out a specific place in the brain that's necessary for opioids to work. Furthermore, rats will self-administer opioids directly into their ventral tegmental area. They don't need their bodies. They just will stick it right into their brain. So that's good. If you give drugs systemically and you put naloxone into the ventral tegmental area, you block its rewarding effect. Right? So ventral tegmental area, and this is a big literature, is necessary and sufficient for drugs to produce reward, for opioids to produce reward. 55% of the neurons in the ventral tegmental area are dopaminergic. And there's good evidence that when you activate dopaminergic neurons, it's rewarding. So the general theory for how opioids work is that they're predominantly inhibitory. Here's a dopamine neuron. This is a GABA neuron, it's inhibitory. I know anesthesiologists know about GABA because it's crucial to your lifestyle. Many drugs work on GABA, including general anesthetics and benzodiazepines. But there's mu receptor on the cell bodies here. There's mu receptors on the terminals. So if you inhibit either the terminals or the cell bodies of the GABA cells, you will disinhibit the dopamine neurons. And that's kind of the canonical model for how opioids produce reward. And it's true, this is work from Alyssa Margolis in the lab, when we actually tested this hypothesis. So you can study a cell electrophysiologically, You can fill it with biocytin, so you can go back to the tissue and find the cell you recorded from. You can stain the tissue with an antibody for GABA, and then you can show that this neuron, retrospectively, was a GABA neuron. And here's a GABA neuron. This is, you can see its resting membrane potential was around 60. And then when you put on this mu-selective agonist, it hyperpolarized the cell. So those cells exist. There also is inhibition of GABA terminals, and these are from cells outside the ventral tegmental area. So in this case, we used optogenetics. We injected a virus that expressed channel rhodopsin, which is the light-sensitive excitatory opsin. It's transported down to the terminals. So we put it into, let's say, the ventral pallidum or the nucleus accumbens or the RMTG, doesn't matter. And then here in the green are the terminals, the red are the cells in the VTA, and you can see the cells are surrounded by terminals. You can shine light on these terminals. You can get a GABA-A response that's shown here. It's blocked by gabazine or picrotoxin, so you know it's completely GABAergic. And then you can show that it's inhibited by DAMGO, that's shown here. So yeah, the terminals are inhibited. So now you have Muse activating dopamine by inhibiting terminals. But then, most recently, we discovered that there was a direct effect of opioids on dopamine neurons, and it was excitatory. And the way we found that out was we patch onto these cells, we show that they are depolarized, and we show that they're depolarized. Here shows a cell being depolarized by a mu-selective agonist. It's reversed by a mu-selective antagonist, right? But it was associated with an increase in conductance, which means that it opened a channel. It was not disinhibition. So there's direct excitation. And furthermore, luckily, we were able to find out that this was due to a calcium current, because there was this snail toxin, omega agatoxin, which selectively blocks PQ calcium channels, and it completely blocked this depolarizing effect. So here's another thing that opioids do, and that's just to summarize the data. So there's at least 3 different mechanisms on 3 different sets of cells that opioids do. They can activate the dopamine neurons by either hyperpolarizing a GABA cell, inhibiting the GABA terminals, or directly exciting the dopamine cells. So there's 3 different possibilities for how different downstream signaling can affect our activation of dopamine and our reward. And the cool thing is that the direct excitatory effect occurs at 2 orders of magnitude lower concentration, which means when you give the drug, it's really the postsynaptic effect that dominates. Okay. We've done that. So in summary, mu agonists are the most effective analgesic agents. It's because they work at multiple sites. The cellular signaling ratios differ for different ligands, and ligands can be designed now to optimize different actions. The mu-opioid reward and dependence contribute to dose escalation and addiction, and we really located the targets that we can use to improve the design of ligands if we get them. So we've got a strategy now to actually, we hope, get a non-addicting but potent analgesic opioid. Take advantage of the design characteristics of the opioid system and the distributed, mu receptor throughout pain modulating and pain transmitting pathways. So we can optimize properties, and I think technically the way forward is pretty clear. I'm very excited about it. Sorry that I'm going to be retiring soon, but there are plenty of people out there that can do that. And when you do that, you will get the Nobel Prize, you will get intellectual property so you can start a company and get an IPO, and then maybe you can afford to live down here. And with that, I'd like to thank the major players here. Alyssa Margolis and Greg Helmstead had done a lot of the electrophysiology. These are people that were technicians in the lab. our collaborators outside, and of course, the funding agencies. And I'll be glad to take questions if you have any.
Howard, quick question, and thank you for that great overview. And it's question's going to pivot a little bit off of this. You mentioned Eric Sun's work in JAM Internal Medicine, and he led that effort. One of the things that he identified in that study was that there was a vulnerability to the likelihood of being, not only being male, but also depression, pre-existing depression, which nicely built off of work that Ian Carroll and Jen Haw did in smaller samples here at Stanford that showed once again that depression was a big vulnerability. Do you have any mechanistic thoughts between that intersection, where that may be occurring, whether there's anything that can be done about it?
Yes. You know I'm an optimist. It's a problem that's really, the technical ability is there to solve that problem. So there's this great study that came out of Roberto Melano's lab looking at a connection. It's basically a basal ganglia connection between a nucleus called the antopeduncular nucleus and an area called the lateral habenula. And what they found is in models of depression, the neurotransmitter in that projection shifts from being GABAergic to being glutamatergic. Now, if you treat the animals with an antidepressant, you restore the ratio. So there's a target. So one possibility is that this depression is actually driving a circuit. And we've recently discovered that there's evidence that that same circuit is driven by pain. So as far as part of the brain's concerned, being in pain is not really different from being depressed. There's a part of your brain that doesn't know the difference. And there's very good evidence that kappa antagonists and mu agonists can at least produce temporary reductions or improvements in depression. Right? So if that turns out to be the case, it's conceivable that there's mu receptors either on the postsynaptic cells in the lateral habenula. We found that. It's not published yet, but it's getting close. So it could be that that's how opioids actually improve depression and relieve pain. So these are not unrelated functions as far as the brain's concerned. So I'm optimistic that we'll be able to come up with something for that. Either a kappa antagonist, mu agonist combo, or a drug designed for a specific set of signaling, intracellular signaling pathways for addiction and depression. Yeah?
Did the mu opiate knockout mice yield any insights into what endogenous opiates are good for? Did they behave or just...
Yes, that is a fantastic question. I can tell you that they drank less alcohol. And it was interesting because the delta knockout mice, they drank more alcohol and they were more anxious, right? But they haven't figured out really how to examine the mu knockout mouse to see if there are more subtle behavioral effects. There must be. There have to be, because the endogenous opioids are all over the place. And if you give people a high enough dose of naloxone, they will start complaining that it's dysphoric. And that's most likely due to some kind of tonic endogenous opioid action. But it would be very difficult to get that kind of information from a mouse. You could, but it'd be tough. Yes?
Accounting for the fact that you're an optimist, how long do you think till we'll have sort of new opiates in our toolkit to treat patients?
Well, if Trevina's lucky, their compound should be on the market within 3 or 4 years, at most. I mean, they already have it in people, so I think that's going to be a big step. There's a bunch of companies that are working on developing kappa antagonists, and there's been at least one clinical trial, didn't work out too well, but there'll be more. So I'm saying that's probably 5, 6 years, something like that. I think we're still very early in the sort of dependence addiction area. I don't think there are any molecules that are out there that have been extensively tested in animals yet. So that's probably more of a decade away.
I just want to comment on buprenorphine. I spent some time with Walter Lynn down in UCLA, and I was from a difference between giving buprenorphine intraoperatively, which I've seen in Europe. You don't see much because it's an operation that's over and you walk away.
Yeah.
Clinical experience, but clinically, buprenorphine acts as a full opioid agonist, and that's what Walter Lynn taught me, and that's what I saw.
Mm-hmm.
Colleagues. And so that's just a point. I mean, I'm not sure what that means. But
Well,
it's
yes.
a partial agonist.
Yes.
The agonist aspect,
Yes.
very low levels of buprenorphine, and when you see it, it's dramatic, but you don't always see it.
That's right.
And so that's one question. The other thing is, do you know anything about anything interesting in the annual cyclist area? Because you talked about beta-arrestin, and I know there's some work on annual cyclist pathways trying to get around some of the cyclists.
Yeah, most of the physiology has been done looking at the G protein signaling, not so much the inhibition of adenylate cyclase. But it's pretty clear that when you inhibit adenylate cyclase, there's a cellular compensation. So now there's an upregulation of the cyclase. Now you take the mu agonist away, and there's this big rebound effect. So I think most people now think that it's a compensatory effect of an increase in adenylate cyclase that causes dependence. To get back to the full agonist, partial agonist action of buprenorphine, that's a really important concept. And the concept is that whether it's a full agonist or a partial agonist is going to depend on the cell model that you use to test it. So it could be a full agonist in some neurons and a partial agonist in other neurons, right? And then the other thing is it a full, how do you say whether a compound is a full agonist? The only way you can do it is if you have another agonist, right? And you found that other agonist produced a bigger effect than any other thing you had. Your drug now is as good as that one. You say it's a full agonist. But if someone comes around tomorrow with another drug and it has a bigger effect, then none of those drugs are full agonists anymore. So it's a relative term, full agonism. I think the critical issue for buprenorphine is, and the data's out there, is it actually produces less respiratory depression at a ceiling. So you've got a plateau effect, and that's why it's a partial agonist for that, and that's good, right?
Well, I mean, if you look at this molecule like the beta-arrestin, you could have addicts who get high all the time, assuming it's still euphoric.
Oh, I'm sure you can get high with buprenorphine.
I'm talking about the beta-arrestin molecule.
Yeah, absolutely. That they don't talk about that.
You still get addicted.
They don't talk about that. Okay.